Okay, so in the interest of time, I'll skip ahead here. I do want to start with acknowledgements rather than ending with acknowledgements. So I'd like to especially acknowledge Wright in the National Toxicology Program, NIHS, which provided some the funding for cell lines that I'll talk about toward the end of this talk, as well as the NIH NCGC, which has provided a lot of essential service in order to do this screening that I'll talk about, and the US EPA that has funded me and the main collaborator of Von Rusten at UNC for several years. And the first one advanced, but this one won't now. Where am I supposed to point at? Pressing this hard. There we go. All right. So before I begin in earnest here, I want to emphasize that my training is in statistics, and I collaborate a lot with toxicogenomics researchers. But I have to have explained to me and re-explained to me the various modes of action and why chemicals are, of particular interest, et cetera. I focus on the statistical side, and these kind of collaborations are increasingly important in this kind of work. So I'll present this as I see it as a statistician. So how should we think about risk for populations in all the senses we've been talking about, but viewing this through the lens of in vitro screening of cell lines? And I'm going to talk primarily about human cell lines. And a lot of the caveats in this kind of work are I think, pretty obvious, but I'll repeat them anyway. One is that you're severely limited in an in vitro environment. You have divorced these cells from the intracellular kind of signaling environments that you ordinarily would have. They may not be the right cell type at all. And then there are sources of technical variation that you might be introducing through, for example, transforming the cell lines or batch effects and other things that are inevitable in this kind of screening. On the other hand, thinking positively, there's a lot that you can do with this, and one of these things is a kind of heritability analysis. So if you know something about the genotypes or familial relationships of the individuals from whom cells are drawn, then you can understand something about variability and heritability. And if you get down to genetic mapping and have sufficient sample size, then you can start to talk about potential mechanisms based on SNP associations, et cetera. Even in the absence of that, you can characterize average response and variations across agents and chemicals if you have some kind of assay, for example, a cytotoxicity, and use that as a means of prioritization for following up chemicals for further study or other agents if you're interested in drug efficacy or adverse events. And finally, you can potentially use these data as in vitro data that further supplements predictive toxicity models. So if you have relatively gold standard toxicity assays, then you can add this into predictive models in combination with something like quantitative structure-activity relationships in order to boost the power for predictive models. So this screening can be relatively cost-effective, but cost-effective is somewhat in the eye of the beholder here. It's not exactly cheap to do this, but there are considerable economies of scale that can arise from the use of well-studied cell lines. So you might use the fact that somebody else is genotyped. Somebody else may have done methylation profiling on the cell lines, and if you obtain them and do the screening, then you can get all of these other types of data that's available on these essentially for free. And I want to emphasize that screening using a relatively few lines, but in a deeper manner, often isn't a good idea, at least in terms of understanding risk and variation of populations. Then you need to understand variation at the population level, maybe both within and between continental level populations. And an added bonus is something that I've been thinking about lately, which is that screening lots of different lines also unmasks variation that you might have missed otherwise. So various kinds of heterogeneity that may be entirely technical in nature only really comes up when you have a massive project and you're looking at lots and lots of cell lines. Otherwise, if you have relatively few, you may not really be aware that they all have a shared batch effect that makes it difficult for you to get the right conclusions. And so This all follows along with roadmaps for toxicity testing that have been spearheaded by National Research Council and the National Academy of Sciences. Getting into the 20th century now, use of molecular information to inform kind of science-based toxicity decisions has come to the fore, especially now the pathway-based views of modes of action and trying to relate molecular pathways to what's going on in terms of toxicity response. And the actual implementation of this through the efforts at the EPA, NIEHS, and partnerships between them have been really important. And the type of work that I'm talking about here falls squarely within this realm of dose response assessment and pathway testing at the population level. Now, a lot of the work that's been done previously has been in the area of pharmacogenomics, cytotoxicity screening of anti-cancer agents. There's a lot of interest in understanding adverse events and variability that way, but many of these principles apply to any agent or chemical that you might look at. So I'm just going to give a very quick and somewhat superficial overview of work that's been done in this area. And the review paper from Howard McLeod and Aline Dolan and others, I think, is a nice summary of the types of information that we're bringing to this, in which we're looking at some kind of pharmacologically relevant phenotype. I'm going to talk a lot about cytotoxicity, but other sorts of measurements that you might get in a cell-based sets of assays. And you want to combine that with sources of or information on genetic factors, vis-a-vis SNPs, haplotypes, copy number variations, along with information on gene expression. And we could add over here in the yellow sort of omics side other kinds of tissue-specific and ephemeral sorts of information that one might get, metabolite profiling, et cetera, as well as things which are a little bit more persistent but would still, I think, fall on the omics side, which would be methylation and other assays like that. And ideally, you want to combine these all together to better understand the basic science of what underlies this variation in the pharmacological phenotypes. And the recent paper by Alison Mautzinger and Howard McLeod and others, I think, is a good example of what one can do in terms of understanding heritability. So they profiled 125 LCL lines, and those were from HapMap and other Ceph pedigrees, I believe, and profiled cytotoxicity in 29 chemotherapeutic agents. And there's a lot of issue of correcting for initial growth rates, which I won't go into, but to summarize here, we have this heritability scale. This axis can go from 0 to, hypothetically, up to 100%. That reflects the amount of variation which is attributed essentially to genetics that one sees in response in these cell lines to exposure to these chemotherapeutic agents. And we see that the two highest are in the order of 65% here. Now, there are a number of reasons why these might be somewhat inflated. It's very difficult to do this estimation of heritability very well in this context. But clearly, you have a ranking of these chemicals, and the ones that are the most heritable are the best candidates to then carry forward to looking at a more fine level on the genome through genotyping and essentially a synthetic kind of GWAS analysis. And even if you don't have related individuals among those for the cell lines, then there's an emerging concept of using genome-wide identity by descent relationships, even among unrelated individuals, in order to come up with heritability estimates. And then you can carry this further because these cell lines have also been genetically profiled. And so there are SNPs along the genome that are available to the investigators, and so then one can do genetic mapping, either by association, which you would typically do with unrelated cell lines, or by linkage analysis, in which if you have smaller extended pedigrees. And so this heat map shows with, I guess you say, the dense line across here in chromosome 7, that there are several of these drugs show mapping evidence in a similar region. And kind of the density of shading reflects how the strength of the evidence. And as is common in these studies, many of these do not achieve genome-wide significance levels. So there's a lot of effort to both increase the power by increasing the number of individuals that are studied, as well as to try to gather secondary information from the mapping information to try to understand whether certain regions or certain types of genomic features are enriched in the data that you look at. And I'll give some more examples about that in a moment. So another group that has been very active in this area includes Nancy Cox and Eileen Dolan. And here's an example in which they were performing direct association mapping. Now, again, this is with non-family-based data, so cell lines that have been genetically profiled but are of unrelated individuals. And so they had a history of doing this on several different chemotherapeutic agents, and then in a more recent paper summarized the genomic features that they saw that tended to show associations. So they have these 6 anti-cancer agents examined for SNPs. Each of them has been tested for association with the cytotoxicity index, kind of an IC50. And they group these SNPs into functional classes and possible function among non-coding SNPs. And then, based on more recent work, had also threw in evidence for expression quantitative trait loci. In other words, these SNPs can be classified according to whether they are predictive of variation in expression at some genes on the genome. And because I don't think this has been described in detail here, I'm going to show a couple slides about what we mean when we talk about expression quantitative trait loci. So an EQTL is a locus, a position on the genome, that's associated with variation in RNA expression. And so this may refer to an individual SNP, especially one that may be causal, but it can also be a broad region defined by linkage analysis. And if you have a microarray, set of microarray data here within individuals and on the order of 10,000 or more expression phenotypes, you can compare them to the order of 10 to the 6th or more SNP genotypes on the same individuals. And of course, this would be on a particular tissue that we're able to profile, but the genotypes are static and thought to be essentially constant for the individuals. And then compare these all to each other. So it's like performing a GWAS for every expression transcript. And so this would be on the order of 10 to the 10 comparisons or more, which is a lot. And there are a lot of issues of computation, error control, and biological relevance. And as I have mentioned before, there are important issues of toxicity, relevance for any particular tissue that you look at. If you have healthy individuals, you can often only get blood cells. But there are a number of other initiatives which will help bridge this gap, such as GTEx, which will gather this sort of information, but from autopsies and cadavers, so that you can get a profile across many different tissues. All right, so what does this have to do with toxic response susceptibility? And my simple answer is that you can provide evidence for or against a transcript-based hypothesis for SNPs or genotypes influencing a toxicity phenotype. So you're trying to get it at the intermediary level of understanding expression as an intermediate phenotype. And so this is a simple model for SNP influence expression, which may or may not influence susceptibility directly. The SNP to expression comparisons, of course, as I said, involve many tests, but the expression to susceptibility involves fewer tests because there are fewer genes on the genome overall than there are SNPs. And this expression to susceptibility testing may involve, or may be thought of as more proximal than genotype. And so in some cases, you may have increased power. But there are a lot of intermediaries that I'm skipping here. The networks that were discussed before are kind of concepts, but if you only can see part of what's going on at the expression level, you don't see post-translational modification, what's going on at the protein level, then it's thought that there may be many intermediary steps between all of these the steps that I'm showing here. And of course, expression is not static, and your susceptibility can feed back to expression, either through disease or treatment or whatever. And So an ordinary GWAS typically tries to connect a SNP directly to disease status or susceptibility as measured, in our cytotoxicity assays. And then an EQTL study will compare SNP to expression. And if you are in the favorable situation that you have expression data on the same samples that you're performing GWAS, then you can combine those two in especially powerful EQTL analysis in which you have SNP and expression data on the same individuals. So all of this would be true for potentially a GWAS study, and it's doubly true here where we have the cell lines. They've been very well profiled, as I'll describe in a moment, and we can look at cytotoxicity phenotypes on many chemicals. Now, back to the study I spoke of a moment ago, in which they were looking for functional enrichment of different types of SNPs among those that were at least modestly associated with their cytotoxicity phenotypes. And the numbers in the table here, essentially, if they're big positive or big negative, they represent an enrichment for that particular genomic feature among those that were showing cytotoxicity association. And these are kind of z-score. And the main story of this paper is that all of the things that you would typically pay attention to for this type of work, for direct functional characterization of SNPs, whether they're missense or nonsense or change, otherwise thought to change proteins, really doesn't have a lot to do with increasing this overall association. And even among the non-coding SNPs, any kind of putative function really is not particularly enriched. But for EQTL evidence, there's a great enrichment. These values bigger than, let's say, 3 or 4 are showing substantial enrichment of EQTL evidence. So the EQTL evidence basically says that these SNPs change the expression of something, either in the particular samples we're looking at or maybe in other published studies. And so I think this has been repeatedly shown for these data as well as for enrichment for disease-relevant GWASs as well. So expression seems to be important, and merely knowing that a SNP is in EQTL influences the chance, in some sense, that it in fact is a disease SNP or is affecting the cytotoxicity phenotype. All right, so a note of caution on this kind of screening that was made, I think, most forcefully by David Altshuler, Mark Daly, and Edwin Choi, which is that this is a difficult process to do in cell lines. If you have as few as a few hundred, then sometimes it's hard to get significant association. And they also found that the baseline growth rates are positively associated with some degree of evidence that one sees in the data, which they attributed to essentially non-genetic factors. In other words, growth rates may be something of a confound representing individual variability that is not necessarily genetic in origin. And so this has led to some concern in the field that maybe a lot of what we're finding is kind of false stuff. On the other hand, the other papers showing the enrichment of various kind of EQTL features really aren't easy to explain in terms of other kinds of confounds. And if you increase the sample size substantially, as has only really recently been done, in fact, you can find what appear to be significant and very sensible associations. So I'm showing a plot here from Alison Motzinger-Reif and Howard McLeod, in which this is a submitted paper, by the way, but they have reported with a larger number of LCLs that are screened for cytotoxic response to temozolomide, which is one of the top 2 heritable traits that heritable response traits that I showed a moment ago. In fact, has a genome-wide significant hit in a already known susceptibility locus that has been widely studied. And so all of this is to say that it's not entirely crazy to be screening cell lines for these traits of real-world relevance. Okay, so now I'll pause for a moment with a summary here. We have epidemiological studies, GWAS studies, which are going on, are routinely genotyped for millions of SNPs, and We have an intermediary that I don't have time to talk about, which is using mouse or other rodent models to try to fill in the gaps here. And now we're proposing to sequence or genotype cell lines and do various kinds of exposure studies on them to serve as kind of a synthetic phenotype to do multiple GWASs with, and to try to tie all these sources of information together. And this fits very well within some recent testing paradigms that have been proposed to try to, again, bring toxicity testing forward in a kind of a comprehensive way, and to identify where this falls within an entire chain of hyperthroughput risk assessment. What I'm talking about here is to talk about population variability estimates as well as to understand the biological pathways that underlie toxicity response. All right, now, in terms of choices of cell lines, one wants to have a lot of genetic variability. This is a graphic from the 1,000 Genomes Project, which has continental variation from Europe and Africa, as well as Asia. And so we at UNC have worked for several years to try to build a pipeline for doing toxicity screening. And so we have a number of tested chemicals which are relevant to the EPA and part of the NIEHS 1408 compound set. Obviously, to do this well requires robots and a lot of automated screening. And this image here shows an image of a plate, essentially with the various chemicals put on it all at once with a whole range of doses. And with high concentrations, you'll see this kind of pixelated map of several of the chemicals showing a lot of cell death, whereas at lower doses you won't. And this all involves a lot of careful work with positive and negative controls. And I would focus on this kind of cytotoxicity profile which is indicative of cell death. Data for these kind of analyses are always challenging to work with. If you're to look at an individual cytotoxic response profile, it's pretty clear what's going on. As you increase the dose, then you're going to be killing cells. And you have to do some sort of curve fitting and decide what kind of phenotype you'll take out of this. Here I have a single phenotype that I'm focusing on, which is kind of a 10% EC10-like quantity, at which basically you have 10% of the cells dying, if you will, and that dose would be a potential phenotype for that particular plated assay for that individual for that particular chemical. There are always issues of outliers, and so we have, for this particular data set, many instances of implausible or outlying points, and so you have to have an automatic way of flagging them and refitting the data. This graphic over here shows that there are batch effects in the data. Those of you who might have seen microarray analysis talks would, I think, be a little bit familiar with this idea that you can have various kind of sources of variation which really have nothing to do with what you're interested in, and so you have to either, through covariate correction or a batch correction, try to align things so that you get rid of extra sources of variation. And I want to very briefly, because I only have 5 minutes left here, talk about the concept of population toxicity versus other notions of toxicity. And so this will involve, at the very least, means and true variability obscured by technical variation. So in a concept here, we have a chemical, and the observed data can fall in a range for those, let's say, the EC10 values that I talked about. This doesn't count against my 5 minutes. Okay. All right. So you have that range of variation that you see, but this has been obscured by technical variation to some extent. So there is a less variable true variability across the population because you have that. The observed variability is that true variation convolved with some kind of measurement variation. So you might have a measurement here, but there's technical variation which is not particularly of interest. So there's some kind of shrinkage that you might want to think about in terms of understanding variation. Again, these will be across cell lines. Not, there's a lot of extra work to understand how relevant that is for free living humans. Okay, but even if we knew this range of variability in our population, then we might want to highlight a vulnerable subpopulation. And I'm kind of illustrating it here to the left. And whatever that limit is that defines the vulnerable subpopulation might be of interest. And this is just for one chemical among many that we're looking at. And so you could rank these chemicals according to that mean response. And here, if off to the left is more cytotoxic, then I guess chemical 4 is the worst. But how about comparing chemical 1 and chemical 3? They're pretty similar in their mean profile, but you'll notice that chemical 3 actually has a more bad, if you will, off to the left is represents higher cytotoxicity, more bad vulnerable subpopulation than does chemical 1, simply because of this variation. And this is something we see in real data. And so the question is, how would you rank these chemicals in terms of the mean or in terms of that vulnerable subpopulation? And so this is, again, very elementary, but something that comes up immediately when you start to do this sort of work. And we also want to mention that the observed variability has this potential to provide some kind of more fine notions of uncertainty factors in risk assessment. All right, so in the hyper-throughput screening toxicology literature, there isn't much data to support these concepts across multiple populations. And so I'll give you an example here of what data really looks like. So we have here an example with 87 cell lines. These happen to be HapMap cell lines. They're profiled by our group, by NCGC, but in collaboration with us. And you can see the gray profiles represent the observed data for these 87 individuals and kind of an average fit through the data. And for each of these 87 individuals, you can fit an individual EC10 estimate, which provides this histogram showing that kind of variability. So this histogram is like that black curve that I showed. And you can see just by eye that there's potential sources of extra technical variability that aren't easily accounted for in a model, and you have to think about as well. And so in the interest of time, I'll very quickly just mention that we have a series of studies that have become ever more ambitious. And the first one involved only 14 chemicals in these 85 cell lines. And the second one involved a larger number of compounds, 240, on roughly the same set of HapMap individuals. And the take-home message for us is that we have very large variation in the degree of variability as well as means of the different compounds. So if I have them ranked here from most cytotoxic to least cytotoxic, you can see ranges of variability that one has with chemicals. And then we've already been able to do a little bit of kind of playing around with genetic mapping with these, and it showed an example chemical here and a typical kind of GWAS pattern. And we can layer into this now RNA-seq data as well that begins to inform how EQTLs might also be used with these sorts of data. And so putting everything in context, all of the studies that we and others have done could pretty well be fit into a small portion of this potential curve, where you have number of individuals on this axis, number of compounds on this axis, and what we want to do is get more comprehensive in the number of individuals here. And so where we're headed with larger numbers of individuals is a more comprehensive population-based view, into which we can also layer on expression, epigenetic data, sequence-based characterization. And so where I'll close is that we now have ramped this up to using 1,000 genomes data, so roughly 1,100 cell lines. And 1,000 genomes is distinguished in the fact that these individuals, or these cell lines, are being, in fact, sequenced, not just genotyped with high-frequency variants, but even low-frequency variants. And as this data matures and accrues, we can actually do sequence-based comparisons to the cytotoxicity assays on something like 180 chemicals now. And having done this, we can, try to take the tenfold default of population variation and fill it in with something which feels more real, and begin to do GWASs as well. This is data hot off the press. I can't name the chemical right now. But these genome scans are apparently showing stronger evidence of association than of course we could get with more than 10-fold fewer cell lines. And I'll stop there. Thanks.
We've got about 25 minutes at the end of the discussion.
Do you want to wait until the, okay. Fred, I think this is really interesting. One of the issues around the measurements that you're making is different measurements are at different levels of biological organization. So cytotoxicity might be a big integrator versus, say, caspase activation might be very specific. Does the fact that you're using an integrator essentially matter in terms of the variability? And I'll sneak in another question that you might be able to answer at the same time, which is all the variability that you showed us has been in one normal distribution. But I think what we all often worry about is a displaced group off to the left. Do you see any evidence of that either?
I'll answer the latter question very briefly first. I think that we do see some chemicals with relatively extreme variation, and this notion of a group off to the left. We see a few instances in which that might be happening, but actually we were more about technical variability and problems with the measurements in that case. So it is a possibility, but until we can attribute it to someplace in the genome, we're a little bit concerned about making broad conclusions. And here we're limited by the number of, basically our throughput, money, and the assays that we can do right now for cytotoxicity. We have done caspase versus ATP in some other previous studies, and I think as we move forward with this, then other kinds of screening for other types of assays are on our plate next.
Thanks.